Good evening, students. Today we will discuss about second year topic, that is phytohormones. So far we are discussing about different types of hormones, of which today we will discuss about cytokinins and ethylene. So cytokinins first identified and crystallized by Skoog and Miller scientists. The term kinetin, actually cytokinin, Skoog and Miller scientists discover cytokinin type of chemical or hormone that named as kinetin, that identified and crystallized and named that as kinetin in herring fish sperm DNA. So these kinetins which are not occur in plants. So kinetins mainly found or observed in animals only. When we observe the derivatives of cytokinins, adenine and N-purpuryl aminopurine. So zeatin also chemically similar to the cytokinin. It is isolated from corn kernel and also coconut milk. So zeatin is also acting as cytokinin. So it is a type of cytokinin found in corn kernel and coconut milk. So cytokinins mainly synthesized in a region where the cell division occur rapidly. So where rapid cell division takes place in the plant body, there only cytokinins being synthesized. So examples of that particular regions: root apex and shoot buds, younger buds. These are the parts where the cytokinins which being synthesized. When we observe the functions of cytokinins, mainly these are help in production of younger leaves or new leaves, and also production of synthesis in leaf, and it also helps in formation of adventitious root. Adventitious roots form in shoot system, and it also helps in overcome the apical dominancy. We already defined the definition of apical dominancy while discussing about auxins. So apical dominance means the lateral buds which not grow fastly when the apical bud is present. When we remove apical bud, lateral buds grow very fastly, that called apical dominance. Cytokinin is also helpful for delay leaf senescence. Senescence means fall of leaf. Okay, and also it also helpful for mobilization of nutrition. When mobilization of nutrition, the leaves do not fall off and the leaves do not get senescence. Senescence means actually aging, getting aging. And it mainly involved in promoting of cell division process. Cyto means cell, kinesis means division. So the major function of cytokinin is cell division in plant roots. The ratio of cytokinins and auxins, the formation of differentiation of callus depends. When cytokinins more, the root initiation takes place in callus, and when auxins more concentration during tissue culture process, shoot initiation takes place. So this is about cytokinin, very important. Now come to the second type of last type of hormone, that is ethylene. So the precursor of ethylene is actually secreted released in the plant body as gas form, gas state, remaining all hormones which are liquid form. except ethylene. So the precursor of ethylene is methionine. Methionine is a type of sulfur containing amino acid. When we observe the functions of ethylene, ethylene helpful for or promote horizontal growth of seedlings, and it promote senescence. Just now I told you senescence means aging. So it promote aging to fall, leaves, fruits, different birds, etc. And also it can be promote abscission. Abscission means fall of fruits, fall of leaves, etc. in the plant body. So ethylene promote senescence as well as abscission process, and it is also highly effective in fruit ripening. Fruit ripening means the younger fruits get maturity. So fruit ripening mainly affected by releasing ethylene, and it is also enhance respiratory rate during ripening of fruits. So ethylene mainly involved in breaks of seed dormancy and bud dormancy. So the other function of ethylene is promotes rapid elongation of internodes and petiole. So elongation of internodes, petioles rapidly, that promoted by ethylene. Ethylene also promote root growth, growth of root and formation of root hairs. So these are few advantages and few disadvantages of ethylene. So that ethylene is defined as plant growth inhibitor. Now the last one is seed dormancy. Seed dormancy means actually sleeping condition of seed. Otherwise inability of viable seed to germinate even in favorable conditions. What is the reason for seed dormancy? There are different reasons. One is impermeable seed coat. When the seed coat is rigid in nature, so the seed will not be germinate. So that undergo dormancy. Example Fabaceae, best example beans, and some of the chemicals which acting as inhibitors for seed dormancy. Some inhibitors responsible for seed dormancy. Example abscisic acid, phenolic acid. These two chemicals which are acting as inhibitors, and low temperature also responsible for seed dormancy. When low temperature present, that seed undergo seed dormancy. Best example polygonum. Thank you.